So good afternoon. Welcome, everybody. I'm Scott Saleska. I'm a professor in ecology and evolutionary biology. I have a joint appointment also in environmental sciences here at UVA. I'm co-director, along with Betsy Arnold, of the U of A's Ecosystem Genomics Initiative's graduate training program called BRIDGES, which stands for Building Resources for Interdisciplinary Training in Genomic and Ecosystem Sciences. BRIDGES is an NSF research and training program, an NRT, and along with Bio5, we are happy to sponsor this important interdisciplinary seminar series, which is starting for the spring semester today. Betsy, my co-director, couldn't be here today due to a sudden change in her schedule. She normally would, but since it's timely, I'm going to take the opportunity to be able to congratulate her in absentia for being named just this week as a fellow of the AAAS, the American Association for the Advancement of Science. This is a great honor, so we're very happy to have her join that group. I'm also really pleased, of course, to be able to introduce our seminar speaker today, Yang Song, who is an assistant professor in Hydrology and Atmospheric Sciences, and who represents a kind of unique pillar of strength of our Ecosystem Genomics initiative here, because she has taken on the very important research task of building mathematical representations that allow us to take micro-scale biologic information, genomic information, about the capacity of microbial metabolism, and translate that into dynamics that matter, or maybe not, that's part of the question, for the cycling of carbon and nutrients at ecosystem scales. And there's only a very few people who've started working in this important area, and Yang is one of them. Dr. Song received her PhD in 2015 in atmospheric sciences from the University of Illinois in Urbana-Champaign. And before that, she got a master's in ecology from the Institute of Botany of the Chinese Academy of Sciences. So it's probably that diverse background, all the way from ecology to atmospheric sciences, that is allowing her to bridge these important disciplines and these important scales. And she's already been recognized before she got here for this work when she was named Outstanding Postgraduate Researcher in Environmental Science for her work with Dr. Melanie Mays during her postdoc at Oak Ridge National Lab. Then she arrived here at the U of A in January 2020, just in time for the pandemic, and just in time to join our BRIDGES program as a founding core faculty member. So we're really eager to hear about her work. So without further ado, let me turn the floor over to you, Yang, where you're going to tell us about the challenge of genomics for ecosystem modeling.
Okay, thanks, Scott. Giving a very nice introduction. Let me first share my screen with all of you. So, do you see my slides? Okay. Okay.
Looks great. Thank you.
Okay. Yeah. Thanks everybody. So I'm Yang, I'm an assistant professor from the Department of Hydrology and Atmospheric Sciences. So today I would like to introduce the challenging of the genomics for ecosystem modeling. So that's one part of my main research here in U of I. So first, Scott asked me to, it's better to introduce how I came to this journey. So basically how I came to this field of the ecosystem genomics. So that I basically, you can see that each step, I basically has a very diverse training background. So as Scott mentioned that I first got basically the environmental engineering, that's from my college. And then I got a master's degree in ecology. At that moment, I researched the methane emission from the plant. That is also a very interesting topic at that moment is around 2006. So that's the first time a German scientist found that the plant can also emit the methane emission. So that is the first research topic actually I started to work on. Then I realized that I know that the methane is a very important greenhouse gases, but I know how that will be emitted from the ground part, but I don't know how that really will impact on the greenhouse process. And the greenhouse gas means that the radiation transfer, how that will really change the temperature condition. So that is actually one motivation that drove me to change my major from ecology to the atmospheric science when I came to the US to pursue my PhD. So my PhD work mainly focused on to develop the Earth system model and better represent basically the vegetation, how in the Earth system model, how that will reflect the interactive with the climate. So that is my PhD research. Then during that time, I also realized that, so because when we try to better understand how the vegetation will interact with the nitrogen cycle, carbon cycle, and how those biochemical cycle process will interact with climate, there is a missing gap that in the below ground. So actually microbial community had not well represented in the Earth system model. So that introduced a larger uncertainty. That is the second motivation that moved me from the atmospheric sciences. After I finished my PhD, I moved to the Oak Ridge National Lab. So I started to work this very interesting and challenging topic, that's the genomics informed soil modeling. So that is also there I start to learn what is the genomics, then how to use those information into the Earth system, into the soil biochemical model. So later with this research interest and experience, I came to here and be part of this bridge program mentor. So I think that is basically my journey. So in summary, I could say that basically the interest-oriented research that make me to change my direction of my research and the direction of my major. But finally, you will realize that all the things will be integrated together, because now a lot of the sense that it is an interdisciplinary research. So that's about my journey to the ecosystem genomics. So now when in my lab, we mainly focus on the three parts of the topic. That first, that relate to the metagenomics, how we better use that information to represent the microbial function in the model. And so I call this as the small bug and also the big rule. And then we're also still working on the vegetation part. So we will better understand the dryland vegetation, how that will interact with this changing hydroclimate, and how we use the remote sensing data, multi-scale different data to better understand the vegetation process. And also I'm interested to look at that the human activity, how human introduce a land use change, for example, the bioenergy, grass production, and also the agricultural management, and how those processes will also change the interaction between the land and the climate. So today I will mainly focus on the first topic. So I will talk about what is the current challenge about using the genomics data to better understand the below ground microbial process and the ecosystem modeling. And then I will show you some of my examples how I try to overcome each of these challenges. Okay, so first let's look at the motivation of the research. So the motivation for me, that you can see the figure on the left, that is basically showing the projection of the global mean temperature over the time by the current Earth system model. This is intercomparison model projection research. So basically they incorporate all different Earth system model, then use that to identify what could be the future climate condition. So you can see here that basically the projection, the global temperature will increase by 2 degrees. So by this, and the uncertainty is around 2 degrees. This uncertainty is actually very large, because if you remember what that the Paris Agreement mentioned, that they target to decrease the global mean temperature by 1.5 degrees. But now even the model uncertainty is around 2 degrees. So that actually motivated us that we need to better understand why various uncertainty come from. How could we mitigate this uncertainty to get the high accurate projection? So then if we look at the source of this uncertainty, the figure in the middle showing that this is the climate and the land carbon flux exchange and how it's changed over the time. And the red one, that is from the ocean part, the carbon exchange between the atmosphere and ocean, how it will change over the time. You can see that compare with the ocean. So basically the climate and land carbon feedback, that still has a larger uncertainty. So that is indicated that we should focus on the terrestrial ecosystem, better understand how the carbon will be exchanged between the land and atmosphere. So this figure basically coming from a paper that is published in 2013. So this is almost the first land Earth system model. So globally, they try to represent the macros in the model, then see how that will impact on the carbon dioxide emission from the land. So you can see that here, this different land, that is means that you use a different model. DSAN and CM4C, that is a very traditional land surface model. They actually don't really have the macros in that process. Then another, this dash line, that basically they introduce the macros in the CM model, and but they also consider that because the change of the climate, some macros, they can adapt to the warming condition in order to change their carbon using efficiency or change how they respond to a different temperature condition. So they try to incorporate this feedback. Then this line, top line, is just don't consider this kind of the adaptation to the warming condition. So just using this different scenario, this research, they found that actually when we project the change in the global soil carbon under the future climate condition, that has a huge difference. So this research indicated us that it is important to better represent the microbial function in the model, understand how microbial will control the biogeochemistry process, how that will adapt to the environmental change. But since this is a 2030, from that time so far, there's still a big challenge for us. Why? Because we know that compared with the vegetation, microbial community is very difficult to be observed. So the basic challenge for us to predict the microbial function in the model, I usually organize it as the 2D, 1D is diversity. So that actually we know the microbial community, they are very diverse. So we don't know this diversity of the microbial community, and also we don't know how that diversity will regulate the composition of soil enzymes and also its ecosystem function. Another D I concluded as a dynamic question. Basically, we don't know how the microbial community will respond to the climate change and how this response feedback will in turn have the effect on the biochemical process. Then the third challenge that is related to the heterogeneity question. So because that we know that globally there are very diverse microbial community species, and they distributed in the different environmental conditions and very response to the different environmental conditions. So for this special pattern, we still don't know, fully understand, and how this pattern will interact with the environmental condition is still a challenge question for us. But later, the good thing is that we found a kind of the data. That is the thing we talk about, that is Omics. So Omics, that is a very powerful tool that actually includes different current biotechnology, which is and with the Omics. So we really together call those different technologies as the Omics, like the metagenomics that sequence the DNA that from the soil, and also the metatranscriptomics that actually extract the RNA from the soil, and the metaproteomics, so that basically identify what protein exists in the soil, and the metabolomics that basically try to understand the metabolic process of the microbial community. We call all those technology as Omics. This figure just showing an example what the metagenomics Omics data means. That basically means if we can get the soil sample, we extract the DNA from the soil sample. It's a little just like today when we do those the analysis, when we test whether we get a positive or negative COVID-19. So we get the DNA extraction, then we do the DNA sequencing, we further assemble it together. So this part you see is an ATGC, that's basically like the coding that of the microbial community. So if we can better understand this coding, we use the annotation, then we can basically know the information about the microbial community related to who they are, means that where they, what kinds of microbial species is there, and what is the structure of this microbial community, and what is the function, means what role they play, and also the what will be the interaction, the relationship between each other, different microbes in this community, and how this community will respond to the environmental condition. So basically, if we can get a soil sample to do analysis for those different omics analysis, that is the information we can get. So far you will feel that, okay, the technology already gave us this information. We should use this to better predict the microbial informed soil biochemical process, right? But that is actually still difficult. So now, in the rest of the time of this talk, I will discuss each of these challenges, what we will face now. I will also discuss one kind of solution, my lab, we are trying to solve it. So the first challenge about the genomics used for the ecosystem modeling that relate to the data complexity and functional redundancy. So if you see the figure here, as I mentioned, this is almost the result we can get from the any omics analysis. But you can see this information actually is very complex because there's so many microbial species in the soil. And they interacted with each other. And you expect it even for one soil sample, you usually will get a huge amount of the data. And also different microbial, they may play the same role, means that they may play the same function, decompose the same organic matter in the soil. That is what we mean that the functional redundancy. So because of those information, it's very difficult for us to use this data to do the prediction. So how could we solve this challenge? One solution I developed when I do my postdoc research, that is, I defined a term called the macro function enzyme function class. So we actually can parameterize the microbial function diversity by grouping all the enzymes that can be indicated by the genomics data into the different enzyme functional class. So here, if you see this figure on the top right, that is that we can classify all the enzymes in the soil based on what chemical component in the soil it will be catalyzed, and at which location of this chemical compound this enzyme will catalyze that. So based on this information, we can classify all the abundant of the soil enzyme just into the several enzyme functional class I denoted as the EFC. Here I'm just showing an example. So we got a soil sample that actually from the Panama, that in this has a long-term fertilization experiment since 1998. So they gave the phosphorus fertilizer here because the tropical region usually the phosphorus limited location. And we got a soil sample from this location. Actually, you can see the data is very huge. Even for one soil sample, we got around 100 gigabase pairs of the genomics data. Then we further understanding what these genes indicated what kind of the enzyme in the soil. We found that it's around 2,000 enzymes exist in this soil. So there's still a huge data for us to do any prediction. So then we extract the enzyme from this, all those 2,000 enzymes. We're only looking for those enzymes that relate to the soil, carbon, nitrogen, and phosphorus decomposition. So that is why we extract around only 180 enzymes here. But this still very large. Then start from here. We just based on the approach I just mentioned, we can classify it as this different enzyme functional class. So here, for example, you see that all the green color part here, that is all the enzymes, we can group it at either they catalyze the carbohydrate decomposition, then this will be one group. We call it as the enzymes related to the carbohydrates. And then if they catalyze the lignin decomposition in the soil, so that will be another group. For the nitrogen one, we can see whether they catalyze the protein in the soil or they will catalyze the pectin. So that's just one approach we use to do this simplify. So then basically we can solve the first challenge. The second challenge here is that if you look at the genomics data, that usually indicated you either told you that what species existed in the soil and the abundance of this species or the abundance of function in the soil. So this abundance that is actually have no unit. So that is another actually the challenge for us to link this to do the prediction of the ecosystem modeling, because that in the any modeling we know that we usually try to either follow the mass flow, means that if we try to simulate the carbon dioxide emission from the soil, we want to see how the carbon mass will change from the organic carbon, then release it to the gas phase as the carbon dioxide, right? So basically we need at least a unit of the mass, but the omics data, they have no unit. Then how we link those information with the ecosystem process? Another challenge here, you see that since the genomics data, that is always at a very fine level, that means at the gene level. But when we talk about any ecosystem process, that usually at a larger scale level. So how we link this different level of the data with a different scale together to understand the question, that's still a problem. So now let's see my solution, how we could solve this problem. So in order to link this new unitless omics information to the ecosystem process, here we develop a kind of the model structure based means that we can link the omics with the biochemical kinetics through the materialist mental equation. So basically what this means that for all the biochemical information that you most of for the biochemical research, they are already in the lab, try to understand for the different enzymes. They do the experiment to test that what will be the chemical decomposition rate of that enzyme when that enzyme try to catalyze a specific organic matter or specific materials. So those information is already there. That's what we call here as a kinetics. Basically, that is some kind of the decomposition rate information we can directly get from the biochemical database. And then we can, from the omics information, we will know that what will be the microbial community composition, and we also will know the enzyme composition. So then we also know that how this enzyme composition will change in response to the environmental change. Those information will tell us that in a soil, what will be the relative contribution of each enzyme in a specific function we are looking for. Let's still take the decomposition of the carbohydrate here as example. So if we think about in the soil, they have a different chemical compound like here showing has a carbohydrate, lignin, protein, and also the different kind of the phosphorus. Then for those enzymes that can catalyze the carbohydrate, its decomposition rate depends on the composition of the enzymes that do this function, and also depend on the kinetics parameter of each of these enzymes. So by link this biochemical database with this omics information together, basically we can quantify here the parameter in the materialist mental equation. So materialist mental equation, that basically is a function that understands how the decomposition rate of organic matter of a substrate will change with the substrate concentration, and also will change with the decomposition coefficient. So that is our approach how to link this unitless omics information with the ecosystem process. So we solve this challenge. Again, there's another challenge we have now solved. We mentioned that the omics information is at a very small level, gene level. How we can upscaling this gene level information to the ecosystem level process. So that is another thing. We realize that actually the omics data can inform that is the life history strategy of the microbial community. So in ecology, we usually use the life history strategy to describe how a kind of the microbial community, they will try to adapt to the change of the environmental condition. For example, this figure I show you here, just an example, you can think about that basically, if there's a three kind of the nutrient requirement for the vegetation, they need carbon, phosphorus, and nitrogen. So then each of this, the angle of the each of this triangle that indicated those enzymes, those EFC that relate to this enzyme decomposition. And so if the CEFC this has a more value, so basically that can catalyze the organic matter carbon, release the more carbon used for the microbial community, so that can mitigate the carbon limitation. That is what we usually call as the ratio, the resource ratio optimization strategy. So microbial community, they throw the competition with each other, then all the time they will try to adapt to the environmental condition. So if a location there has a nitrogen limitation, they usually can allocate the more enzymes that relate to the nitrogen decomposition. Then if there's a location has a high phosphorus limitation, they will allocate more enzyme related to the phosphorus decomposition. So that's what we call this as a life history strategy. We realize that at the gene level, basically this can indicate this strategy. So this is one of our earlier research publication. We realized at the gene level, you can see this red color line that indicated the specific enzyme. Each this number here, that is like the ID of an enzyme. So you see that if I show it as a red color, that means that under phosphorus deficient soil, you will expect this enzyme concentration usually higher than a soil without the nitrogen deficient. So that is you can, if you summarize this figure, basically we can see that those enzymes relate to the phosphorus decomposition that will be increased under the phosphorus deficient case. In the meantime, those enzymes, they catalyze the lignin here. Carbon part will be decreased because they try to allocate more resources to get the phosphorus from this phosphorus limited soil. So then this is at the gene level. We develop a numerical solution so that we can upscaling this gene level response to the basically the ecosystem level. So we now, instead of use each of these enzymes, we define as this different EFC, as I mentioned, the enzyme function class. Then we can statistically summarize that for each of these functions, either that relate to the carbohydrate decomposition or the lignin decomposition, how that will respond to the change of the phosphorus ability. So here again is actually this figure on the right is just follow the same same result what we see at the gene level. We also see that those enzymes relate to the phosphorus decomposition. They actually increase under the phosphorus limited condition. So that is our approach, how to link this gene level environmental response to the ecosystem level response. Is this clear?
Can you, Yang, hi, can you clarify the axis on the bottom? I'm a little confused. The positive side says decrease and the negative side says increase.
Oh, yeah, here, this one, you mean the figure on the right, right?
Yes, on the bottom, yeah, the words say decrease, but the numbers are positive.
Yeah, so here depend on ID. So I gave some notes at the bottom to indicate that. So if it's on the right side, so that basically means this value will be decreased under the phosphorus rich condition. Then if it's on the left side, that will be the increase under the phosphorus rich decomposition, rich soil. So basically, if we see this value is positive, so that basically means that this value will be, and if there's a nitrogen phosphorus limited, we'll expect this enzyme will be increased. Is that clear? No. So here actually what I'm showing here, this is a log. Showing result here is a log EFC concentration under the basically the phosphorus under this the rich condition divided by the same EFC under a phosphorus limited condition. So if it's so that what the y here, that should be the log log that is the for example, let's just choose the C lignin as example, that C lignin under the phosphorus limited condition divided by C lignin under the phosphorus rich condition. So this is the ratio.
Yeah, okay, so it's greater than 0, that means greater in real. Log 0, that means 1, greater than 1 in real.
Yes, yes. So then if this value is negative, so that basically means that under the phosphorus limited condition, they will decrease this value. Is that clear?
I think we should, so there's something about the explanation that is confusing.
Yeah, I think I should give the definition here.
Yeah, that's it. Never mind. Go on.
Okay, so then let's look at the third question, challenging here. So when we try to put the omics information into the model, that usually will hear the two kinds of the voice. One voice that usually coming from biologists and ecologists, because we know that the microbial community, they are important. They really control a big role in the soil biochemical process. So we always promote us that put this process into the model. Then another voice will coming from the Earth system modeler, because they know that Earth system model is already very complicated. So they would worry about the question that is actually not a question only for this research, they also the question for all the different components of the Earth system model. What will be the model complexity and uncertainty, how to get this balance? So in summary for this question, it means that we try to use the microbial omics data to represent the diversity of the microbe community. But how much we need it? What's the level of the microbial community diversity is enough for us to predict the ecosystem process, and so that we also don't need to introduce too much of the parameter, introduce too much of the uncertainty into the model. So in order to answer this question, we try a kind of the sensitivity analysis. We can use our enzyme function class that approach just classify the enzymes into the different way to indicate whether that will be the low functional diversity or a moderate functional diversity or the high functional diversity. So here, if you see the figure on the left, that is the most simple version. Here, we don't even consider what the chemical compound in the soil. So we're just assuming that all the things that only have the carbon like the lignin cellulose as a one group, then we can group all the enzymes that catalyze that as a one EFC. And then if we compare this with the figure in the middle, now we further classify the soil organic matter based on their chemical compound. So those enzymes only catalyze the carbohydrate, this will be one EFC. Then those enzymes, they only catalyze the lignin, this will be another EFC. So then if we go to the right part, this is the most complicated classification. Not only consider the chemical compound, we also consider basically the polymer set of that chemical compound. So we can follow the carbohydrate example. You see here, in order to decompose the carbohydrate to those small monomers so that the microbes can use that, actually that is this 3-step. So once the liter, those carbon coming from the vegetation to the soil, that's still very larger chemical compound. So those enzymes first just cut some internal bond of this larger chemical compound. They make it a little smaller. Then another group of enzymes will continue to work on this, further catalyze that to the smaller one, until that probably they only have the change that to the monomer carbon. That is since the microbes can really eat it. So you can see that the teamwork of these 3 EFC, they together will catalyze the decomposition of the organic matter. So we just use this way to define these 3 functions, 3 different kind of the diversity. Then we see that what kind of the decomposition process of the organic matter it will be introduced. So this is just a summary about this different model. So you can see this table here. I summarize that if we try to simulate the microbial CP ratio, that is a carbon phosphorus ratio, all the microbial carbon dioxide flux, or short-term soil organic carbon the carbon content or the long-term organic carbon content in response to the warming condition. Just if a model try to simulate each of this process, which model maybe give the best enough solution result. So here we always compare each model's result with the observation. Then we pick up the one which can give the very fit the observation condition. If both all the model, for example, if the low diversity model and high diversity model, they can both fit the observation very well, we will choose the low diversity model because that will be more simple. So that is how this figure come out. So you can see that if we talk about the CP ratio of the microbes, especially under the control case that basically in the if there is a phosphorus limited soil, you will also always see that here the model which we consider the high diversity of the microbial community. Also, when we consider this dynamic change of this microbial community, the enzyme function to the environmental condition, this is what we denoted here as a HD, they will perform best. But for the soil where don't have the phosphorus limited, even the low diversity model, that's enough. Then for the other, the microbial carbon and short-term soil organic carbon and even the microbial carbon dioxide fluxes, I think we see that the low diversity model that's already enough. When we try to understand that how the long-term organic matter will be changed in response to the warming condition, then we see that the high diversity of the microbial community can give the usually the simulate the light soil carbon loss compared with the low diversity of the model. And also, if we further consider how this microbial community will respond to a change of the environmental condition, that also will further change the result. So the figure here just showing you example, you see that if we look at the future 100 years, how the organic matter will be changed in response to the warming condition in this Panama soil, then you will see that basically when we use a high diversity of the microbial community to represent that into the model, that will project the less organic loss, carbon loss. So basically here, this gives us an idea that it's not means the too complicated will be always better. It really depends on the question and where we try to understand this question. So if we are looking for try to understand the long term and the soil biogeochemistry process into a phosphorus on the nutrient limited condition, usually we need the represent the high diversity of the microbial community. But if we are only look at the location where usually not the nutrient limited and or during the short term, low diversity of the microbial community can be represented into model, and also that doesn't require a lot of the parameter. So that's basically our solution try to solve this challenge. So far,
Yang, just quickly, just to understand the figure on the previous page.
Okay.
So it says control at the beginning.
Oh yeah. So
that says, go ahead. That's without any phosphorus addition.
You mean that the lamp trend figure, right?
The lower left figure says control with a flat line.
Here, this control means that this is the soil with the phosphorus limited. So basically, that's the same as the column, first column of this figure.
And then at some, does that mean that phosphorus has been added there?
Yeah, there's
starting around 2030 or something.
Yeah, so we don't give any fertilizer, then just like it's running. So we will expect the phosphorus will continue to limit it in this location.
But so does the figure capture the effect of phosphorus addition, or is it just all of it's the control one?
So this is just the control one.
And then when the lines start getting changing, that's just due to the different parameterizations of the model.
Yeah, so that basically starts when the environmental condition introduce a signal here to make it change. So at the very beginning, the environmental condition the warming condition doesn't change the below ground a lot. And
just so there's some of those lines show the Coleman model that has the microbial, the genomics information in it, and compared to the non-genomics information.
Actually, all this has the genomics. So here L here means they're low functional diversity.
Yeah. Okay.
So all has the omics here.
So is there some, maybe you're getting into the comparison between the genomics enabled versus no genomics model?
Yeah. So this just to try to understand how much of the genomics informed diversity we need to introduce.
Okay. And but you believe one of those more than the other ones, or they're just part of the variability?
So I think here that this conclusion that if we answer the question in the location where don't have the nutrient limitation, we could just use a low diversity, represent the low diversity of the microbial community. But if we're looking for a location where really has the nutrient limited condition, then we need represent the high diversity of the microbial community.
So the green is low, so we need the red line instead of the green line.
Red line or the black line. Black line, the difference is that further consider the microbial, this enzyme functional can also change. So means that we parameterize this ratio, results ratio optimization series. We consider this a life history strategy.
Yeah. Okay, go on. Okay. Sorry.
Okay. Yeah. So far you can see that what I introduced based at a set level. Now again, there is a big challenge we have not solved. That is the environmental heterogeneity of the omics data. So basically, we know that when we get the data from the microbial community, they are very diverse. So we go to a different location, you will expect that the composition of the microbial community will be very different. So the approach I just mentioned, whether it can be also worked in other location, we don't know. So one Chinese question here is that the general enzyme functional class, as we have seen in that Panama set, will be also found in other location, and how does this composition abundance of this different enzyme functional class will vary across the different diverse environmental conditions. So in order to answer this question, we actually develop an environmental factor informed machine learning model. So basically, this figure showing here that where we got the genomics data, we try to collect all the genomic publish the genomic data from the genomics data set like the GGI and also the NMBC. And so you can see we found for this analysis, we found around 28 sites that cross the US domain. So that include around 277 samples. And it represents 8 typical ecosystem types, include both forest, grassland, and cropland. The good thing is for this data, we also have we got this sample from the different depths. So basically from the shallow 5 centimeter until to the 115 centimeter. So that allows us also to look at whether model the general enzyme functional class can be identified in the different soil depths. So using this data, we also pair the environmental data with this genomics data. We collected the, for example, like the climate condition, the precipitation, and also the temperature condition. And also we classify the above ground vegetation information like the NPP and NDVI. All this will tell us that how much of the litter will come into the soil. Another will be the soil texture, like the sandy and sand, shade, and clay fraction. And then we also look at the chemical component of the soil, like the soil organic carbon and also soil serum ratio. One thing is that we don't find a lot of information about the soil phosphorus content. So instead here, we also introduce a soil order because soil order that sometimes can indicate the long-term phosphorus availability in the soil. So that is like here showing this different soil space, that's the different soil order of the data we have collected. Then for some order, you will see that actually indicated that the phosphorus limited. For others, that will be the non-phosphorus limited. So that is the environmental data we collected and pair that with the genomics data. Then we built several different kinds of the machine learning model. So this research mainly done by my student Fan Changpeng is also a participant student of the bridge program. I think what he learned from here is also help him to working on this project. So the machine learning model, what we try the random forest model, and that's basically like a parallel machine learning that integrated the different decision tree, like this each one here that try different decision tree. Then we put it together, try to do the prediction. Here, what's the this prediction, how to integrate it means that we usually consider environmental factors. Like I just mentioned here, that is a climate condition, soil physical process, soil biochemical process, and also the vegetation properties and the measured soil depth. We use all those information as a predictor. Then we see that if we have those information, we can, for some set, we also have the genomics data, whether we can train the machine learning model to link the relationship between the environmental condition and the genomics condition, means what is this enzyme function classification. So here, the prediction here that will be the relative abundance of each enzyme functional class as we classified. So that is how this research is done. And then in order to try different machine learning model, we also construct that is called the gradient boosting model and also the adaptive boosting model. The difference between this model with the random forest model, basically that is like a sequencing search of the best solution. What happens that it will first looking the using the one decision tree to check try to classify those enzymes into the different functions. Then they will find some data, maybe when they do the test, they see something that cannot be well do the classification. Then they further focus on those amount of data, either give the high weight or just introduce that into the next training. So step by step, so this way we can try to improve the accuracy of this machine learning process. So that is the our approach. So here, the first question we want to understand is that whether the machine learning model can be used to identify the enzyme functional class. I think the answer here is yes. So if you see the result here, that is the result related to all our testing data. So here, each column here showing you the different machine learning model, and then this EFC, total EFC NP, that basically means that we grouped all the EFC that relate to the carbon organic lignin cellulose as one group. This is all related to the organic nitrogen deposition. That's related to the organic phosphorus deposition. Then, based on our specific classification of each of the enzyme function classes, this is also the specific EFC we defined. So we want to see that whether model could use the environmental condition to predict the relative abundance of each this EFC composition here. Then the value here, that is the model performance. So if this value is larger, it means the model did a good job. So it can predict this the abundance of this functional class. So if you see the distribution of this figure, is also indicated that actually we here run the model by several times, use a Monte Carlo approach, so that we try to make sure the choice of the training data and the testing data will not affect on the result. So you see our result here is very converged. It means that the model has a very consistent result. And compare here different model, you see this adaptive boosting model basically gave the better performance. So from the sense part, we can conclude here that you can see that the machine learning model can predict the enzyme function class involved in the organic carbon, nitrogen, and phosphorus decomposition. But if you let's look at the details of this model performance, you may realize here for the monomer phosphorus and also for the monomer nitrogen, those enzymes, the performance is relatively low compared with the others. So we think about this either because here in this prediction, we have not introduced a microbial biomass as a predictor because this data is very limited. This is probably one reason that introduced this, basically this prediction is not looks good. Another is also coming from the availability of the whole machine learning data, because we know that in order to do a good machine learning research, usually we need more data to train the machine. But since you can see now we have only around this 277 data, so that's probably too limited. Another question here that is actually a sense question. We know that how we classify this enzyme function group, we basically group those enzymes that do the similar job in the one class. We define it as an enzyme function class. But we know that the omics data can be has a very high heterogeneity. Means even in the very similar environmental condition, it's likely possible some enzyme will be not there, some enzyme may be there. So the definition of this group may be slightly different. So here, there's two challenges we also try to solve. Whether we have enough omics data to develop this machine learning prediction. Also from the sense part, so can model capture those fine scale difference in the enzyme function composition? So even they under the similar environmental condition. So in order to answer this question, we introduce another approach here called the data augmentation. This concept is borrowed from the machine learning research, how they use to identify different image. So like the figure here showing an example, usually if we want to identify this image is dog, then but we want to have more data to train this machine. So what they do that they will change the angle of this figure. So you see that this four figure here, they actually generated based on this original image, but just this dog in this image has a different angle, but they are still the angle, still the dog. So we will use those data to train the model, see if model could still identify this is a dog, not the cat. So that is what a data augmentation usually use the image identification. We can borrow a similar concept here to identify the enzyme function class. For example, here we
Yang, I just want to encourage you to hopefully get to the end soon because we have less than 10 minutes before.
Okay.
We'd like to have a little time for questions too. So.
Okay. Yeah, I think I will skip this part since if you are interested, we can discuss it later. So basically, so this augmentation approach. Now you can see we can basically here to make the data from 277 until to the 3000 data. Then you will see the model result actually can improve a lot. This is our original result with the data augmentation. This is basically the without this is the with this data augmentation. You can see the basically the model perform a good job. Then We also found that there's a general enzyme function class we can actually classify from all the locations, those with the sample we have got. So this is the definition of those enzyme function class we defined in the Panama. We actually found a similar enzyme function class in all other different soil samples we got in this research. Then the most interesting thing is that we want to see what event condition dominated the abundance of this different enzyme function class. So this figure is showing for those each of these enzymes that relate to the organic carbon, nitrogen, and phosphorus decomposition, which environmental factor is important. From this figure, you can see those very light color. For what we concluded here, that is the soil carbon and also the soil saline ratio. They are very important factor to predict the EFC that relate to the carbon and the nitrogen decomposition. But for the phosphorus, we see the soil depth, elevation, and also soil nitrogen. They are a very important factor. This actually indicated that we can even use a machine learning model to identify the organic carbon amount if we have the genomics data. So basically, we can use the machine learning model to do the two-way prediction. Then, if we further look at the details of each of the enzyme function class, some more interesting things we can see that the soil saline ratio is a very important factor to control the enzymes related to the monomer nitrogen and also relate to the phosphorus mineralization. Then organic carbon, that is also important for predict the EFC for the protein decomposition. And we also realize that the precipitation here, you see, this is very important for phospholipid decomposition. And by the temperature, that is very important for predict the EFC for the protein decomposition and also the inorganic nitrogen transformation process. And the other important factor that relate to the location.
One minute warning, Yang, before we have to stop.
Yeah, I'm basically,
okay.
Yeah. So then use this model, basically we do this prediction and we can identify the enzyme function class as the basically the US domain. So that is a summary I would like to put here. So basically, this is all the different challenges I have mentioned. So I show you an example how we try to solve each of these challenges. There's still something we are working on, like here, the gene function expression and the ecosystem process. And also, we know that the uncertainty will coming from a different sequencing approach and also examining annotation, and how the physical process will further affect on the microbial kinetics. So this basically the following research my lab is working on. We are also continue to travel those challenges. So that's all about my talk. Sorry for running a lot of time. So if we still have the time, I'm very glad to hear the questions and comments from all of you. Thanks.
Yes, thank you so much. There are a few questions. You want to speak? Jen asked a question first.
Okay, it's
Jen Lee is a postdoc in our Emerge project who's actually trying to use a different model to get similar kinds of approaches as you.
Okay.
Jen?
Yeah, I think it also is similar to what the question next, Sarita, and also it's the similar question to you, like Scott. But my question is, so what is the standard of the good when you compare this low diversity model and the high diversity model?
Okay, so one standard here that is compare with the observation. So actually, I don't show the comparison with the observation. We have the observation for the microbial CMP ratio and also the biomass and carbon dioxide concentration, all those information. We try to compare each of the version of the model with those observation. Then if it's compared, we use a statistical analysis to identify whether it's good or not. If both model perform the same very similar kind of this statistical analysis result, we will say both model can be works. Then we will choose the one which has the low, which the simple one. So that is how we define it as good or not. Is that clear?
Okay. Yeah, that's clear. Thank you very much. And also, I really like your knowledge about the complexity versus the uncertainty. And when you introduce this high diversity to the models, is there a large uncertainty associated to it? And how do you balance, like how do you compare with these uncertainties? Do you think it's worth to it to add to the more diversity to the model?
Yeah, actually, that's a very challenging and good question. As this test I did, that base is still only about geochemistry model. So at that level, we use this test to see at least for each kind of the question. And we see that if we introduce a more complexity of the process, it's not improve the result. We will not use that. That means always that we introduce more parameter. When more parameter is introduced, always there will be more chaos of that model, right? So more uncertainty. So that's definitely the true. So that's why I think here the summary for this part is that we choose look at each of this different diversity approach, see which one gives the already enough to give the best result compared with the measurement. We will use that version. Then the next step, we really need to put this into the larger scale model integrated with the above ground part, means that how the above ground vegetation has interacted with this one. And also another important thing here, I don't mention that once those mass spectrometer data, now it's very useful for us to identify the organic matter chemistry process. That can help us to constrain that what level of the diversity of the microbial we need to introduce. Then we can integrate those information together to help us to mitigate the uncertainty related to how much of the diversity we introduce into the model.
Yeah, thank you very much.
Okay, thank you.
Thanks, Sarita is leaving, but just to ask her question too. In addition to the question of how good is it compared to data, is how different is it in the predictions of the future? And you started off with the Viter et al. Comparison that showed that including microbial functional representations could make a big difference for the future, but you didn't answer, have you answered that question for your model? Does it make a difference in the future between if you run the non-genomic model with compared to the genomics model?
Yeah, so that's a research actually doing now. So basically compare with the future first, we don't know what is really future happen. That's the only things we can compare with the future calibrate projected by other larger scale model. So that is why I actually now introduce this machine learning process. Then we can use this data into the larger scale model to do this task. So then we can directly compare with what the measurement, what the current larger scale model already do this projection. Then we see when we introduce the Omics data inform the microbial process. How?
But what about Panama? Just Panama? Does it make a difference in the future for just this site that you did the simulations?
Yeah, that's also a strategy. So we have not put the Panama directly into the larger scale to compare with that. That is.
No, but forget about the larger scale for the moment. Just ask for this scale, for this site. Would it make a difference? Does it make a difference?
So
if you use the microbial, the genomics enabled model versus the non-genomics model?
Yeah, so Panama only found not very long term, but at least there has a global, not a soil warming experiment. We did a not very accurate analysis. We see that it at least can capture that trend. But as you mentioned, I think you ought to see that without introduce any omics, we actually put even we have to put the all information into a set level model, then to consider with the without any omics and with the omics. That is the things I'm working on actually. Is that answer your question?
Thank you. I think we are at our time limit, so we should probably stop and thank Yang for her seminar, and welcome everybody encourage everybody to come back in 2 weeks for our next seminar. Which do we have somebody, or are we still? Yeah.
That's the one we're still waiting on. Every other one is booked.
Okay, fine. So we'll have. I think that, there's a website, or is it up on the web yet, Heather? The site schedule.
It should be live next week.
Okay, great. Okay, thank you, Yang. Thank you, everybody.
Thank you, everybody.
I'll see you next time and see the ecosystem, the Bridges students in a moment.
Okay, see you all. Bye.